Thanks, Dennis, for a great introduction. And it has been a little bit of time since I've spoken to you. And I was also a little challenged because Shirley, I think, is going to tell us about great stories about natural products at next lecture. So I hope you come to that one. Today, I wanted to share with you kind of some of the things that are happening in Florida and how we're trying to fit into those and some of the collaborations that we're bringing online and some of the more recent research that we're doing. So let me see if I can make the slides go forward. Florida has made a pretty large investment in life sciences or biotechnology research in the last number of years. They have created, I guess, if I understand the history of this, after 9/11, there was kind of a crash in the economy because things like tourism weren't taking off very well. And at that time, our governor was Jeb Bush, and he said, you know, we need to diversify, we need to have other things, and what are some of those things we can do? And certainly life sciences, which is drug discovery and understanding of the biology of disease and things like that, biotechnology, is a growing area. And there are a number of groups, for example, in the Cambridge area, there's a life sciences cluster, in South San Francisco, San Diego, Research Triangle, and these are all very successful at building the economy of the areas that they're in. So they set up what was called the Florida Innovation Incentive Fund, which would provide funding and incentives for various groups working in life sciences to come to the state of Florida and to build research programs that would augment what was already here in the state. They also started a program in the Centers of Excellence, and that was more at the universities. We could all compete, and we could apply for grants and then get a $10 million package of funding to try to advance our life science programs. There was very recently, what's been set up is the Institute for Commercialization of Public Research. And that's a group that's trying to help those of us who find interesting discoveries take them the rest of the way or get an investor interested so that they can make it to the clinic. And so that's a new group that's just been formed to help us academic scientists. There's also now the Florida Opportunity Fund, which is actually a venture fund, which the state is giving all kinds of incentives to venture capitalists to come and invest in the state of Florida. Because they have identified that although we brought a lot of groups in now that are doing great research, we still don't have that venture capital to really take companies and make startups and make it go. So the state has really invested quite a bit in life sciences. So some of these investments, and I'm going to tell you right away that I searched the web and I tried to get numbers, and there's these reports from the state government, this Office of Program Policy Analysis and Government Accountability at the bottom, that give us some of the numbers for what the state has put in. Where it's a little harder to kind of get the numbers is what the local incentives have been to different groups to come to Florida. But here's the laser pointer. So the first group that really came, 2003, was when they were kind of courted to the state at Scripps, Florida. They ended up down in Jupiter. They're part of the Scripps Research Institute, which is in San Diego in the La Jolla area. World-renowned group for doing life sciences research. They got an investment originally of $310 million, and that's actually paid out in a certain period over a 10-year period. They'll get their last payment, I guess, in 2013. They also had a lot of incentives from Palm Beach County to build in Palm Beach County. The Burnham Institute for Medical Research, which you'll hear a little bit more about in terms of a joint project that we have going on with them, got $155 million in state of Florida general funds. And then somewhere around $89 million, I wrote plus because I think there's actually some venture capital that's kind of associated as an incentive for them as well to come to Florida. And they're in Lake Nona, which, if no one knows where that is, that's actually in Orlando, right next to the airport. And my colleague there, Greg Roth, you can look out and see the airplanes taking off from his office. It's actually really neat to see. Torrey Pines Institute for Molecular Studies is right here in Port St. Lucie. And they got about a $32 million package from the state. And this is a number I'm not so sure about, but about $50 million in local incentives to help build the building. And we know that they were here on our campus for a while, and they're now really growing, and now they're in their new facility and adding a lot to our local economy. Very new is the Max Planck Florida Institute. This Max Planck is this very famous research institute that's primarily in Germany. This is the first one that will be in the United States, and they'll be located in Jupiter, immediately adjacent to the Scripps labs. They got $94 million from the state and about $87 million from Palm Beach County to come there. And so they're not actually on site. They've hired some of their main scientists, but I think they're still just building. They're going to take over the building that the Scripps Group used on the Florida Atlantic campus in Jupiter for some period of time till their building is built. And also in Port St. Lucie will be the Vaccine and Gene Therapy Institute. And from what I understand from reading the web, they've hired their top scientist, and they have about a $60 million package from the state and $53 million from Port St. Lucie and the county. And so they'll be starting up, and I think they're going to be in the Torrey Pines buildings till their new building is built. So if you add it all up, it's over $1 billion in state and local incentives. So there's really an investment in this life sciences here in the state of Florida, and that's pretty exciting. So it's kind of early for most of the groups to really do an accounting of how these groups are doing, but there has been, in October 2009, there was a report, once again by this Office of Program Policy Analysis and Government Accountability, that came out that analyzed whether Scripps Florida should get more money and get the rest of their money. And of course, they were found that, yes, they were in compliance and they were doing very well. Their facility was completed in February 2008, and they brought in over $129 million in grants to this, to their research program there in Florida since 2003 when they first relocated. They currently have 280 full-time equivalents. So what that means is a few people are part-time, but if you add up the total number of jobs, it's at least 280, which was their goal to have by this point. And what I think is really impressive in terms of economic impact, in the fiscal year 2007-2008, Scripps Florida had an estimated economic impact of $168.5 million on the state's gross domestic product, and it sustained an estimated 1,327 jobs. So if you give a $300 million investment, that's just 1 year they've come back with that kind of return to the economy. So I think what we can see is that maybe this has been a good investment of our taxpayer dollars, and that as these other groups who've just, I wrote that Torrey Pines here just finished their facility in September 2008, and Burnham just got into theirs. It was finished in April. They had their grand opening in October. That eventually we're going to see really this kind of economic engine coming from this, and that it's actually, I think, quite a positive thing. One of the other programs I mentioned were these centers of excellence. And so when those first came out, it was in 2003, 2004. They were grants of $10 million to a university, and the university could then invest that in their own life sciences or any kind of technology program. 2 of those were in life sciences, and the first was the Center of Excellence in Biomedical and Marine Biotechnology. And it was awarded to Florida Atlantic University, and our group was actually a major part of that. And a major part of the whole program was the discovery of natural products from the sea. And just one little number for that, and they only have reports through 2006, and I don't know what the current is. The $10 million investment through 2006, and that was only 3 years after the inception of the grant, had returned $29 million in grants to Florida Atlantic University and its partners on that. So that's somewhat successful. We were able to get a lot of infrastructure, new equipment, modernize, and allow us to be more competitive on our NIH grants. The second one is the Center of Excellence for Regenerative Health Biotechnology, which is at the University of Florida. They built a facility that allows them to do manufacturing of large biological molecules that are used as drugs using good manufacturing practices. So if you're a small biotech company and you want to take and do some testing, you can get your compound made there at University of Florida in this lab. So they have an ongoing program of production of things in that lab. And they've returned about $24 million in grants received through 2006. And I don't have any more recent numbers than that. So once again, the Centers of Excellence helped us build infrastructure. Now, how does HBOI's biomedical program fit in? Well, we actually started many, many years ago, back in 1984. Our group was kind of called the Sea Farm Project. And we had a sister company that was a sister biotech called Sea Farm Inc., which was the drug development arm. So the project would find new drugs from the sea, and the little company would develop those and take those to the clinic. Unfortunately, the stock market crashed in about 1989, and along with it crashed Sea Farm Inc. But luckily for us, Sea Farm Project continues on. The mission of the Sea Farm Project was to discover drug candidates, lead compounds, or molecular probes from marine plants, invertebrates, and microbes for the treatment of human disease. And the focus was really on deep water habitats, because Harbor Branch has a unique tool, the Johnson Sea Leak Submersible, that allows us to go where other people have not been able to collect organisms. And by going there and collecting unusual organisms, we get unusual chemistry. For our program, much of our program mission is still today the same. We also have a program in biofuels, but most of our work is still in the drug discovery game. So we always show this slide. Shirley might show it again next week. I don't know. She's shaking her head no. But nature is an important source of medicines. I like to remind people of that. Natural products are small organic molecules that are made by plants or animals or microbes that don't have a role in primary metabolism. They don't keep the cells alive, but they presumably keep the whole organism more competitive and allow it to survive in harsh environments. There was a study done by Gordon Craig and David Newman, who were both, well, David's still at the NIH as the head of the natural products branch. Gordon was the former head and is now retired. But they did a study and showed that about 62% of the small molecule drugs or agents that are approved have can be traced back to natural products. So they're either the natural product themselves or an analog or derivative, or the natural product was used kind of as an inspiration for synthetic organic chemists to make better compounds that make better drugs. Examples of drugs you might have heard of: aspirin comes from bark of willow or birch, morphine comes from a poppy, penicillin, revolutionized medicine, comes from a fungus, lovastatin, the first of the statin drugs ever found, comes from a fungus, adriamycin, a very important anti-cancer drug, and Taxol, another important anti-cancer drug from a tree. So nature remains a great source of new compounds. So we actually have a marine natural products success. I've put this slide up before in the past, but for those who might be new, this is a beautiful little tunicate called Actinosidia turbinata. You can find it at the inlet. You can find it on pilings in some mangroves here in this area. Throughout the Caribbean, it's quite common. It makes a compound called actinosidin 743. And actually, the first discovery that this little tunicate had a compound that would cure cancer was back in 1969 was the first report. It took till 1990 for us to determine this rather complicated structure. And it took many more years for it to get approved. And it's only approved right now in Europe, but it's approved for soft tissue sarcoma and just been approved for the treatment of ovarian cancer. So there is a marine natural product that is now marketed, at least in Europe. We hope someday we'll hear it's actually marketed in the United States, but that seems to be taking forever. But it does kind of show that marine organisms, there's some hope. Now I want to kind of digress a little bit and talk about a grant that we've received that kind of fits into this life sciences arena. And I want to start by talking about our Harbor Branch collection of marine organisms. So we have a very large collection of marine microbes. So each one of these test tubes has an isolated microbe that comes from the marine habitat. This is Peter McCarthy's marine microbe collection. And here's an example of one of the beautiful microbes, if you expand it up and look at it. And so this is a resource for drug discovery. We also have a large collection of macroorganisms. So by that I mean ones that you can see with your eyes. So soft corals and algae and sponges and tunicates and things like that. And this actually shows part of the voucher collection. And then this is our big, large freezer. And there's a whole bunch of milk bins. And in every milk bin, there's all these organisms. And they're just preserved there at about minus 15 Fahrenheit. Almost the temperature, I think, that we've been having recently here, it feels like. We probably could have just left the samples out. It would have been fine. But anyway, so we have this very large resource. And so one day I was up, I gave a little talk at the Burnham at their brown bag series, and I showed slides like this. And one of the researchers said to me, well, you know, you really have a gold mine of chemistry there. I mean, think of all the compounds that are in all of those. And I thought, gold mine, yeah, that's a good catchy term, gold mine. So we tried to get a number of different grants to start mining the gold mine. Oh, there's one of our lovely sponges, Liatomation. We talked about that last year. And we weren't very successful at getting grants for a while. Well, we have a lot of grants, but not to just go in and get chemistry out. I wanted to go back and say that these are actually different marine organisms, and wherever you get different marine organisms, you can actually get different chemistry. And these are the way chemists draw chemical structures. And by the end of the talk, you're not going to want to see any more of these, but I'm going to show you a few chemical structures as we go through. But the general rule is wherever you have biological diversity like this, you get chemical diversity. And chemical diversity gives you the opportunity to find new medicines. So this is actually what's called a thin layer plate. So a thin layer plate is one type of chromatography that we can do. And what I'm showing here, and it's kind of blurry because they look kind of blurry, is we spot extracts of many different organisms across the bottom. They're all of the same genus, a genus of sponge called Batzella. So you can get the idea of diversity within even one genus of sponge. Each one of these little spots, blurry spots, is one or more chemical compounds. So the idea of this slide is to show you that from each of these sponges, there's many different compounds present within each of those sponges. And if I were to send this to Esther Guzman, who's our cell biologist who tests for cancer, anti-cancer activity, she might tell me this extract is active, but I don't know which of these compounds it is. So I have to figure that out. But the main point is extracts are very complicated and they have a lot of compounds. I want to step back one more thing and tell you, just so you know when I use the word extract, what I mean by that. Some of you have heard me speak before, so you may remember. But if you made a cup of coffee this morning, or you made a cup of tea, you made an extract of coffee beans or tea leaves, and you pulled out things like caffeine and flavoring components, which are all natural products. So when you make an extract of, make a cup of coffee, you're making an extract. We don't use hot water, we actually use ethyl alcohol to pull out our compounds, but it's the exact same kind of process. So traditionally, how we've always found interesting natural products, as Dennis says, interesting natural products that might be active is we use this kind of process we call bioassay guided fractionation. So I have that across the top up there. And so what it is a whole series of iterative chromatography testing, and this shows one kind of column, and once again we put a solid in there, a solid called silica gel, for example, and we put our mixture of extract of all the things on the top, and we start pouring through different liquids, and those liquids will end up with the compounds being kind of attracted to the liquid or attracted to the solid we put in there. And when they are attracted to the liquid, they begin to move down the column. You see that blue band there, and eventually they end up in this flask at the bottom. We put them into many different flasks, and we separate our compounds. That's one kind of chromatography. Not very high resolution chromatography, but it's one kind of chromatography. So we do chromatography, we test it, take the active fraction, more chromatography, test it, back and forth, till eventually we get a pure compound. And this process can take weeks, it can take months, and in really difficult cases, it can take years. So this is not a very fast process. Now, I've told you that we have all these new groups that have moved to the state, and some of the things that they've invested in are things like this. This is the Burnham, their new laboratories up at Lake Nona, and this system here is their high throughput screening system. So it's ultra high throughput screening. And it's a whole series of robots. Each one of these units, these blue things, is a robot. There they are, 1, 2, 3. This is up and functional now. And all these other things around the edge are incubators or plate readers or liquid handlers or all the things to allow them to automate the bioassay process. So they don't really have people. In our lab, you go there, and our automation is that we have really great research associates and assistants who actually do all the assays manually. They pipette everything, and they grow the cells, themselves. And in this case, all of this is kind of done by the machine. Interestingly, I want to point to this guy here. This guy is Steve Vasile. Now, Steve Vasile is the manager for this facility, and he actually postdoced. So as soon as he finished his PhD, he came here and he worked with Dr. Peter McCarthy on our, at that time, high throughput system, which was nothing like this. It was a couple of little machines, and learned how to do that and went on to industry and then on to the Burnham, where he runs this whole system. I want to point out that when we talk about ultra high throughput testing, they're testing 1 million samples in a 16-hour workday. So they program it all up. It's all computer run. He's sitting there doing some of the programming. And then it just runs 1 million different test points in 16 hours, which is really amazing because I don't even want to say what we do compared to that, but it's nothing like that. But if we want our library of materials to be compatible with people who can test 1 million things in a day, or let's say it's even 500,000 things in a day, we can't go through that back and forth thing. Because what happens is they'll bring up an assay, And they may have it in that system for, I don't know, a week, maybe. And maybe they'll keep it back in the lab somewhere for some period longer than that. But if we have something that takes months, you know, they're on to the next thing. I mean, they'll leave us in the dust. So it's not really practical for us to be testing those crude extracts with groups like the Burnham. And they're actually testing some of our stuff right now. So, and we want to work with them because they're part of what's called the Molecular Probe Screening Network Center for NIH. And the goal of those labs is to figure out what every protein in the human body does by finding a small molecule that interacts with each of the proteins. And that's why they're testing a million things a day in order to find a molecule that interacts with every protein in the body. So it's a really huge project at NIH. It's part of the roadmap project. So what we've been doing for a while as part of what's called a National Cooperative Drug Discovery Group grant that we're on with Phil Cruz at UC Santa Cruz, Bill Fennicle at Scripps, Bill Gerwick at Scripps, and Novartis up in Cambridge, Mass, is creating what we call a peak library. I'll show you a picture of what peak library really means. But in that, we make an extract, we do a simple solvent partition, and then we use this highly sophisticated machine called the Combi Flash to actually do the chromatography for us. It's not a very sophisticated machine. It doesn't have really high throughput, but it does actually separate things for us fairly well and allows us to get a lot of things in our library. We're actually getting a new one of these coming up soon, and I'll talk about that grant in a minute. These are examples of when I'm talking about peak library materials. So this is a crude extract, and these are actually HPLC chromatograms of different, so this would be a crude extract. Each one of these little peaks we're looking at is the UV absorption of a different compound. So in the crude extract, you can see there's lots of different things. Just like from the thin layer plate, you can see all those little dots. So that would be a mixture. And from the Combi Flash, we can get fractions that are greatly enriched. So this one is mainly this compound, has a little bit of stuff here. This one has this stuff I don't like to look at, but then it's got these major compounds. And then this one has a few little things, and then these major compounds. So you can actually trace it up. So if we test this in the Burnham system, maybe we'll only have one step of purification left, if even that, before we'll actually be able to determine the structure of the compound that's active in their assay. So rather than testing this and having to get each of these separated, we can test these subfractions and get there much faster. So that's what we call our peak library, is something like this. So I think everybody knows that we have this stimulus package that has come out, and you know, people like it or they don't like it or whatever. Personally, I love it because we got some money, so we have to be happy. The NIH had something called the Grand Opportunity Request for Applications. And when you read through it very detailed, you found that creating national resources was one of the things, as long as you were going to create some jobs and spend some money in US companies. So we put together the gold mining idea that we would take our collection and make a very large peak library, which we can then work with groups like the Burnham and et cetera, to actually really rapidly find novel natural products that work in assays we would never have conceptualized or could never possibly do in our own labs. So the grant we received is 2 years. It's about $1.4 million. Came to our program, and it's to create a library of natural products compatible with labs like that at the Burnham. And we actually have agreements with a lot of different groups to test the library that we currently have and the expanded library. Because it's a stimulus package, we have to do things like create and retain at least 6 jobs, and those will be here at Harbor Branch. We're also having the opportunity, one of the things we put into it is a kind of a challenge for young people as you go and you get your college degree in, let's say, chemistry. And then you want to get a job in chemistry, but most of the industrial people, they want somebody with a couple years experience. So you know how to really work in a lab, not just in a lab you know, that's artificial in a university. It's actually a real life lab. So we actually are going to be hiring a few people who are just out of college, and this will be their first real lab job, and they'll get that experience. So that was a positive thing for the grant. We're also going to have quite a few summer students, and those can be from, there's age limits at FAU, but they can even be high school, you know, junior college, et cetera, can come and have summer jobs with us, working and learning how to work in a lab. We're purchasing equipment from US manufacturers, and I hadn't really thought of this, but I have some friends who work for companies that make scientific equipment. And just like everyone else in the economy, they had really been having a hard time. They have salespeople, they have people developing new pieces of equipment, et cetera. And if they weren't selling any equipment, they were really, they were suffering. And now through this stimulus, and there's a lot of money through NIH that's going for equipment, we're actually buying equipment from people, and things are moving again, and they're able to keep their people on staff. And I think, the stimulus really it's taken a long time to happen for some people, but our grant only came through in September, really October 1st. So, I mean, we've already bought one major piece of equipment. So I think it takes a while, but it's going to happen. And ultimately, the goal of the grant is to create this national resource for natural products drug discovery so that we can send it out to a lot of different people who have different ways of looking for treatments for cancer, et cetera, and find new, hopefully, drug candidates. And this is actually another part of the grant. We get to buy one of these. It's a Waters auto purification system. It's been used by industry quite a bit, but most academic labs don't have the money to buy one. But we were able to get it through this grant. Waters is a US company, so that worked out well. And it allows us to do mass and UV. So that's molecular weight based upon the molecular weight of a compound, and UV is based upon its UV absorption. Directed purification to give us pure natural products. And this is a research part of the grant to figure out how well this works and whether we can really adapt it. And it will automate our process and make it more efficient. In addition, there it is. It's up in our labs right now, all in boxes. It arrived right before Christmas. Our group has just gone through the process, as many groups here at Harbor Branch are, of moving to new laboratory space. And so we've just moved to the new Marine Science Building. And the labs are now, I'm happy to say, 90% back in order, but things like this are not set up yet. So soon that will be set up. But we're happy to see a new piece of equipment. So now I'm going to kind of illustrate a project that we've done that has involved our colleagues at the Burnham, at least at some level on that, and bring it back to this whole kind of life science cluster, hub, corridor, whatever we want to call it. And I'm going to call this the Afro-Callisti story. So these are all a type of sponge called hexactinellid sponges. And hexactinellid sponges live primarily in the deep sea. I don't know that any of them actually live in shallow waters. One of the things that they have in common with each other is that they have very little tissue, they're very fragile, they're very brittle, most of them, and they're very difficult to collect by ways like trawling or something like that. They would just fall apart in the trawl and break into a million pieces. And they have these siliceous or silica spicules that is pretty much what makes up most of the sponge, and there's very little tissue in these. And we'd collected a lot of them because we worked a lot in deep sea habitats, but we hadn't really seen any that were active. And then we got one that was active. And it's this guy. And it's called Afro-Callistes beatrix, and it's these little, look like little glass vases. They're very fragile, and you can see it's actually growing on one of these deep coral reefs. And we actually collected this on our Center of Excellence cruise in 2004, right off Fort Pierce. 40 nautical miles off Fort Pierce, you can go to 2,700 feet of seawater, and you can collect this sponge on these deep coral reefs. I think the next thing I have is a little video that we're going to show on the collection of that sponge. And I want to thank John Reed. I stole some of his footage. John's in the back. And Brian Cousins for helping put this together. So that's the Seward Johnson. It's out in the canal. That's them launching the sub from below. It's getting dropped into the water.
Roger.
It's making me dizzy. It's swimming around. And so this is actually the sponge. It's not the best footage in the world, but these are them. They're kind of vase-like, and they see all those little yellow protrusions are zoanthids that live inside of that sponge. So they're collecting it. And they're quite abundant, actually, out on the reef track, that deep reef track off Florida. And it's kind of funny that we only got the first one in 2004 that had activity. So they're collecting the other one that was right there. . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . . So they line that, that's called the clam scoop that they're using to collect that. Oh, we'll get it soon. They are really fragile, so you have to be kind of careful how you collect them. So those were the ones we worked on first. And so now this is footage from just this last summer on a NOAA ocean exploration trip that John Reed went out. We needed to get a little bit more of the sponge to work on some minor metabolites. And while they were out there, John, I stole this footage because it's just so amazing. And you weren't giving a talk, so I stole your footage. Anyways, look at how these are little failure reefs, and John can tell you more about it at the end if anyone has questions. But I think this was one of the most amazing ones in that it has almost 100% live cover, and it's just an amazing reef that's out there.
15 feet wide.
You can just see how big that is. And I just want to point out that these areas, due to work that John has done, have been I don't know if it's finalized, but they're being protected as marine protected areas because these are incredible habitats for fish and many other kinds of organisms. And if you trawled over the top of that, that's very brittle and fragile. You'd just destroy it. So they're now being protected.
There in the videos, Chasion, some large Ophelia thickets, 2 meters.
So this is a much better image of what the Afrocalistes looks like. That's one that doesn't have the zoanthids, and it shows them collecting it. And I think John brought back about 14 or 15 different samples, and we analyzed all of them. Did they have the chemistry we wanted or not? Now they're using the clam scoop here to collect it. It's kind of hard to see. But you'll get to see it as it raises up, and you'll see how frilly, and it has these fingers, and it's really a pretty sponge.
The time is 18:30.
You can see all the little frilly fingers, and etc. So that's Afrocalistes that we're working on. And there's the sub coming back on deck. So the question is, why are we interested in this particular sponge, Afrocalistes? Well, we have a grant to work on finding new agents that are active for pancreatic cancer. And some of you may know that pancreatic cancer is one of the ones that has less than a 5% 5-year survival rate. And there really are no good drugs for pancreatic cancer, and it's usually diagnosed very late, and so it's very difficult to treat. So this particular sponge actually wasn't the most potent thing we've ever seen, but it gave good inhibition. And we also had a summer student, and there's a picture of her coming up on another slide, and she's actually was a high school student, she's now at Dartmouth, from St. Ed's, who came to work with us. And so she assayed this in an assay called the sea urchin embryo cell division assay. And it was very potent. It inhibited 100% at a fairly high concentration, but it was 100% cell inhibition of division. And so here's this assay. What we do is the first part of the assay is really fun. We all get to go. We don't get to do a lot of field work. So the first part is we all go to the beach, and we go snorkeling off the reef, and we collect the urchins, which is always the most fun. Then we come back to the lab, and you can spawn them by just injecting potassium iodide. You get the sperm and the eggs, and you mix them together, and then you wait about 1.5 hours, and you get this cell division. So here's the cells dividing, 4 cells, 8 cells, and it's synchronous. So that's what we call vehicle control. That's normal cell division for these embryos. If you have something that blocks cell division, you get something like this. So there's no cell division. They're all just sitting there. They're not doing anything. And so some of our really interesting compounds like discodermalide, dictiostatin, our new compound liadermatalide, they would all show this kind of response in this type of assay. And one of the advantages of this assay versus our cancer cell lines is that for students who are only here in a 10-week period during the summer, they can do one of these assays in an afternoon. The cancer cell lines take 72 hours, so it's about a week turnaround for a cancer cell line assay. So for students, this is a really great assay. They really like to go to the beach. So the purification, and here's Diana. So this is Diana Pector, who's now at Dartmouth. But she came in and was working with us, and so she extracted it. So she added ethanol, ground it up, and got the extract. And then she did something called partitioning, and that's just like, think of gasoline and water. You just shake it up between the two. It's not really gasoline, and you get your compounds might go into the butanol. Dry that down. Do vacuum column chromatography, and when she assayed those fractions, she found that they were active in that assay, the sea urchin embryo assay. And then she did some more, used that Combi Flash Companion, that little machine I showed you, and did more chromatography. And one of the compounds was pure right at that point. Unfortunately, while she was here that summer, we weren't able to test it because all of a sudden it was the end of the summer, the urchins weren't spawning, the tests weren't working, and so she went ahead and started to work on structure. We couldn't actually test it. And now is where it gets really, if you don't like chemistry, go to sleep. I decided I would, since I'm actually a chemist, I was going to bore you all with some NMR spectroscopy, my favorite thing to do. And maybe some of you want to come and solve some puzzles with us. But this is actually what's called a proton NMR spectrum. And most of you have heard of magnetic resonance imaging. This is just the method without the imaging. And we don't have to, we put our sample into a little tube, not like a whole person into a giant magnet. But what we're looking at here is a proton spectrum. So these are hydrogen atoms in our molecule. And each hydrogen atom gives a signal based upon how much energy it takes to take it from a lower energy state to a higher energy state. And if you want a more detailed description, I can give it to you. But I don't think anyone wants to know more than it goes from low energy to high energy. Ones here take a lot of energy. Ones here don't take as much energy. And there's just rules that all the chemists learn for interpreting, you know, if something absorbs here, how much, you know, what might it be? And this is just an expansion of this part of the spectrum. Oh, I also wanted to point out that sometimes you can see that some of these little lines, instead of being singlets like this, we call that a singlet, they're split into multiple lines. So like there's one's 3 lines, and this one is 3 lines as well. So we call it a triplet. And that has to do with the proton on adjacent carbon. So if this is a proton, that's a carbon, this is a carbon, that's a proton, they'd be something like that in the structure. So that's a carbon-13, so that's another nucleus that we can look at, and organic molecules have lots of hydrogen atoms and lots of carbons because it's kind of almost the definition. You add oxygen, nitrogen, phosphorus, and sometimes sulfur, and you've got an organic molecule. So that's the C13 spectrum of her molecule. And what's kind of weird about it was that there was all these weird signals that didn't integrate properly, and it was very confusing, and I have to give Diana credit for sticking with me on the structure on this one, because it was a more difficult structure to do than we would have thought. This is a spectrum that's really important, or a type of spectra called the COSY. And I'm going to walk you through how we interpret it. And it's a little hard to see, but here's the triplets. There's the triplet right there that I showed before. And I'm going to call that, well, first we look at this diagonal. So all COSY spectra have what's called a diagonal in them. And wherever you see a dot, or a series of dots, that's off the diagonal, so it's not on this line, that means that a proton there is coupled to another proton in the molecule. How do we figure out which one it's coupled to? Well, if that's proton A, we draw down and go further to the diagonal, then we go across, and we go back up, and we figure out that A is coupled to B, and there's a partial structure for this molecule. And so the chemists in my lab have all this fun. They get really complicated spectra, and they sit and they figure out A's next to B, and B's next to C, and C's next, and it's actually a really fun puzzle to do. But that's an important type of spectrum. The next one I'm showing you is a little more complicated. It's an HMBC spectrum, or heteronuclear multiple bond correlation spectrum. And here it has proton up here, so there's my proton spectrum again, and there's that carbon spectrum. And this one is telling me which protons are a couple of carbons away in my structure from each other. And I'm going to show you one example of how we would read this. If we go down from that resonance right there, that singlet, down, and we see a dot, we come across, and we say that proton has to be a couple of carbons away from this. It can be 2 carbons or 3. And we have this partial structure in our molecule, and so we would look and figure out what each one of those dots on the spectrum means. And so it's actually kind of a, if you like puzzles, this is kind of the ultimate puzzle to put together one of these structures. So if you're a puzzle solver, come see me. So the structure of the major compound, we do it really fast. We draw it like this. That's what we get from COSY. That's what we get from the first part of HMBC. And that's what we get from the last part of HMBC. So that shows us the final structure of the molecule. Now it's interesting because if I go from this nitrogen here to that nitrogen there, that's a compound that's been known for a while. It's called molechiamine. And that compound, or that part of a structure, is in almost 100 different natural products. So they have different things on either end, or they're dimers or whatever, but that part of the structure is in it. But the interesting thing is that all of those other compounds come from a type of sponge that's totally unrelated to hexactinellid sponges. They're all from Virongids except for one, which comes from an Agilis sponge. So it's like, what's going on? Why would this hexactinellid have this compound? It's unexpected that it would be there. So that's the first kind of interesting thing, is like, why would a hexactinellid have a compound that should be in Virongid sponges? It's only interesting to people like me, but it's an interesting question. So the next interesting thing about the structure, and when I first presented this at a big scientific meeting, people went, what? That can't be right. Anyways, was that we have a methyl group at this position. This is actually an adenine. It's part of one of the nucleotides that make up DNA. So something like this is in DNA. And there's a methyl group on this position, and it's always here in nature usually. Almost anywhere it's here. And there's only a few synthetic reports of ones that have a methyl group there. There's not even very many compounds. So that people were questioning me, like, are you right about that? I was like, well, I think I'm right, pretty sure. But that was a strange thing about the structure. And then the final thing that made it different from other compounds was that it has, on this end, on this nitrogen, has what's another methyl group, and it also has an acetate. And that actually makes it kind of live in two different, what we call tautomers from each other, which was what made the structure difficult to do. But there aren't any others that have both functionalities or anything like that in the series. So that's kind of unusual as well. So we think it's an interesting structure overall. So as Dennis says, chemists always say it's an interesting structure. I like it. Anyways, so what does it do? We had originally seen that it was active in some pancreatic cancer lines and things like that. We tested it against a whole panel of tumor cell lines, and I'm just going to draw your attention to these ones. It's not so active in these two. It's not bad in that one. This is a colon line, and it's about 7 micromolar. Miopaca is a pancreatic line, about 26. This is a drug-resistant line. That's an ovarian line, 34, panc, 22. And this is a breast line. So we actually see, it's kind of moderate activity. We have a lot of things that are nanomolar, but it's still in the range of, for example, 5-fluorouracil, which is a drug that's used to treat cancer. So it's actually better than 5-fluorouracil. So, it's interesting. But what's really interesting is that all of those have something called a mutation in a protein called P53. So what is P53? P53 is a tumor suppressor gene. And it's a tumor suppressor gene that in about 50% of all cancers, there's a mutation in that protein, which makes it so that that protein is not functional anymore. And it's sometimes called the watchdog of the genome. And its real role, and this is very simplified here. There's like a million things in the real diagram. But P53 is here. If you have DNA damage, P53 kind of sets in play a whole system that says, go fix the DNA before you go on and divide into 2 daughter cells. We don't want to pass on mutations. So P53's role is to kind of, check and say, well, the DNA is messed up, so go fix it. If it can't be repaired, then it should induce what's called cell suicide or programmed cell death, apoptosis, and kill that cell, which will then make it that the mutation doesn't get passed on. After the DNA repair is done, it goes on to cell division. If P53 is not functioning properly, then the cell division goes on, and mutations can be passed to the daughter cells. And that can eventually lead to the formation of the tumor, is if you keep having bad mutations that get passed on and then can go to the cancer. So all of the cells that we're active against have mutations in P53, so that might actually be advantageous because we would hope maybe that the afrocolistins will actually show selectivity for cancers that have a p53 mutation. So patients who don't have that associated with their cancer, this wouldn't be any good for them. But if their tumor does have the p53 mutation, maybe we can get some selectivity. Now we are planning on working with a colleague at Florida Atlantic University who has what are called paired tumor cell lines. So they have one tumor cell that has what's called wild type p53 or normal functioning, and then others that are mutated. And we want to see that ours is really active in the mutated but doesn't really kill the normal. And so that's the next step that we're going to be doing. Hopefully we'll see that, and then that'll be good news to take it forward. Now, where does the Burnham group come in? Well, they came in because partly that question about the methyl group, you know, I was pretty sure. But the way to really prove it is to have somebody synthesize your molecule. And so Greg Roth, this is Greg, looking quite unhappy there. He's the head of the MedChem program there. This is a very serious picture for him. And this is Daniela Divlanska, who actually worked with us as a postdoc, but is now a staff researcher at the Burnham. And there's another woman named Jennifer, whose picture I don't have, so you have to imagine Jennifer right here. She has short, dark hair. They went ahead and they did this synthesis of afrocolistin, and so proved that, in fact, our structure was correct. And not only that, but they made a lot of material so that we can now move forward with some more advanced testing in animal models that will happen, hopefully, at the MD Anderson Cancer Center, which is another group that's up at, in Orlando, and that we have a collaboration with. Now, not only did Greg make our compound, but he's in the process of making what we call a library of compounds. So the general structure is up there, and he wants to do things like change, you know, the length and the type of amide that's coming off here, change whether these are bromines or not, change the length here, change the type of heterocycle that's out here, and come up with a library of compounds that will test. We'd love to see the potency get better. It's only in the micromolar, it's okay. I'd love to see us get to a nanomolar level compound. And that's what MedChem people bring to the table, is this kind of ability to take what we call a hit and turn it into a lead and into eventually, hopefully, a drug. And so that's the collaboration that we have going on with Greg's group. He has made a few. So far, the natural product's still the best. But, and that's because Mother Nature is a wonderful synthetic organic chemist, and she makes the one that works the best and then keeps that one. But hopefully, as we go further, we'll end up with some that are even more potent. So I'm going to go back to, we had seen that sea urchin embryo active compound originally, and that's one of the reasons we were really intrigued with this, because we like to find compounds that block cell division. So it became another summer. We had another summer student, Michelle Bousquet, who is a student at FAU. She just graduated from FAU, is looking to go to graduate school. Maybe she'll come here. She did more testing on the fractions and found that the activity was not in afrocolistin. It doesn't block the cell cycle, but it's in fractions 2 and 3. And we now know the structure of compound 2, but I'm not ready to tell you what it is yet because we're still doing all the testing and we don't know how far it's going to go. We've done a little bit of testing in our mammalian cells, so our cancer cells, to see if it blocks cell division, and we haven't really seen the response we want. So this summer, we're all going to, unfortunately, have to go back to the beach, and we're going to have to go snorkeling and collect some more urchins so we can do some more testing. It's really a tough life, but somebody has to do it. I bet the students do it. I don't get to go, but I did go once. Anyways, so we're going to do more work, and we know that the activity is in here, and there's some really trace compounds with some really complicated structures, and so that's the work that's ongoing in the lab now. Oh, and I was supposed to show that those are the actives. So acknowledgements, Diana Pector, Susan Sennett, Michelle Bousquet, Pat, Tara, Esther have all worked on the isolation and first testing of afrocolistin and that series of compounds. Greg, Jennifer, and Daniela did the synthesis. John, collections and taxonomy of the organism, and Peter generally helped with some other testing on it. And the Gertrude E. Skelly Charitable Foundation has funded the summer stipends for our students and has done that for quite a few years. And then a variety of funding agencies that are supporting the research that we're working on. And I think with that, I can say thanks for the attention. And if you have questions, that's our group, our whole research group. Please feel free to ask them. if you like.